one approach to curing HIV is to try and eliminate virus that's hiding in reservoirs. The virus that's hiding is invisible to the immune system, and you need to make those cells visible. And we can do that by activating the virus. It's a strategy that's been around for a while called shock and kill. Problem with previous strategies is we were using drugs that weren't specific for HIV. They were drugs that were activating lots of genes, not just the HIV gene. But what we're doing in the amfAR grant is delivering a drug that's HIV specific. The details of it are a little bit complicated, but it works because it has to recognize HIV sequences or the genetic code of the HIV before it's activated. So what we make mRNA that codes for that drug. We wrap the mRNA in a fat bubble, exactly like you get for your vaccine. But the next challenge is to get the fat bubble to the reservoir. And we're using ways to do that, putting signals on the fat bubble that direct it to the reservoir. So it's pretty cool stuff. It's very early days. And it's a lot of technology that we have acquired actually over the COVID period that we're now applying to HIV. We're doing that all in test tube models at the moment. And next stage is doing it all on cells from people with HIV outside the body. And the third stage is testing it in a mouse model. The first grant I received from amfAR was in 2006, which is a very long time ago. 17 years ago. I was at the very beginning of my career, really, as an independent researcher working in HIV cure. And what was amazing about that grant was that it was given to me on a really good idea. Not much what we call preliminary data, experiments proving that the idea could work. It was just a good idea. And that's a little bit high risk, but most funders won't, especially in Australia, don't fund good ideas. So that amfAR grant was very transformational for me. The good idea worked. We developed a way of studying the HIV reservoir in a test tube model, which wasn't possible before, and the rest is history. And then I've subsequently received quite a bit of money from amfAR, who have just played an incredible role in driving the HIV research agenda forward, but particularly in HIV cure, which has been such a major priority for amfAR. Well, I'm involved in lots of projects across the Doherty. The main areas I work in are HIV cure, HIV hepatitis B co-infection, people that are infected with the two viruses and what that does to hepatitis B, and of course COVID. But I'll just talk a bit really about the HIV work because that's of high interest to amfAR. And the two main areas of work are killing the reservoir through activation or through specific kill drugs, and boosting the immune response. So another major theme of our work is trying to take advantage of drugs that have been developed for cancer that enhance your immune response against the cancer, and whether we can do the same to enhance your immune response to HIV, to one day allow people to stop taking antiviral therapy and let their immune system take over. And the main ways we're doing that are using drugs that basically reverse immune system exhaustion. So in HIV, even when people are on treatment, their immune system is a little bit exhausted, left over from before they ever received treatment. And we're trying to reverse that exhaustion using these cancer drugs, different ones that are injections, ones that are tablets. And again, amfAR supported that project. It was specifically looking at a group of drugs called immune checkpoint blockers. The most famous one is anti-PD-1, or licensed as Keytruda. Lots of people know it because they will have friends or colleagues that might have had their lives saved by this drug, particularly for skin cancer and other cancers. And so we developed, that was a large grant from amfAR, where we looked at these drugs ex vivo, so outside the body, in cells and people with HIV on antiretroviral therapy, in monkeys, and also analysed samples from people with HIV on antiretroviral therapy who also had cancer and therefore were receiving these cancer drugs. So that's a very big program. We've just started the first study of anti-PD-1, that cancer drug, in people with HIV on antiviral therapy who don't have cancer, who are otherwise well. So we're going to really learn a lot about how that drug works and what it does to the exhausted immune system. Yeah, I don't think the expense should deter us at the moment. We're in discovery phase right now. We need the breakthrough scientific advance that's going to cure HIV. And if it's super expensive, it is still going to be a major breakthrough. And then the next step is we adapt it, refine it, and make it cheaper. And that really is the story of antiviral therapy, to be honest. When antiviral therapy first came on board in the mid-90s, it was super expensive and only rich countries could afford it. But it was so cost effective. And we developed a whole new structure for how drugs are priced and therefore their availability in low and middle income countries. So we shouldn't let that deter us. And in the specific area of gene therapy, there's scientific reasons for why gene therapy will eventually become cheaper and more accessible. And the main reason is that at the moment, most gene therapy studies require you to take cells out of the body, manipulate them with gene scissors. In the case of HIV, you might use gene scissors that recognize the virus and put them back in the body. Very expensive, very high tech, very difficult to do outside of a very sophisticated medical centre. But there are now advances in gene therapy, and one of those actually is the technology I spoke about earlier, mRNA wrapped in a fat bubble, that can deliver gene editing tools. In fact, that's what we're using in the amfAR grant. And that's in vivo gene therapy, meaning you get an injection, and if you can give instructions to those gene scissors to get to the right place, you just bypass a lot of the expensive, complicated parts of gene therapy. So I am actually very optimistic that the science will deliver much simpler solutions for gene therapy, and as a consequence, it will be cheaper. But we mustn't stop the innovation because it's expensive. We're just not at that stage at the moment with cure. We need a breakthrough innovation in the science, and then we deal with the issue. Well, there are several ways that people are trying to cure HIV, and one of them that's in the most advanced and now in clinical trial in people is what we call combination immunotherapy. So it uses a mix of strategies to both boost the immune system so that you can get long-term control of the virus in the absence of treatment. And at the same time, some of these approaches also target or try and eliminate the reservoir itself. And certainly in monkey studies, certain combinations of immunotherapy, drugs that activate the immune system and stop it being exhausted and target the reservoir, look pretty good, meaning that a percentage of monkeys in one study, up to 50% monkeys, were actually cured. We're now moving into those clinical trials in people, and it's looking promising. There's a signal, it's early days yet, and that a subset of people, maybe a third, maybe a half, seem to respond to the combination treatments and keep their virus under control for the duration of the study. We don't know whether that's going to be indefinitely, but for the duration of the study, which usually has a follow-up period of about 6 months. But that's only one strategy, which we call combination immunotherapy. Gene therapy is using a very different approach that may not require a combination because you have a much more direct way of slicing out the virus or boosting the immune response. What will get to the clinic first we don't know, but based on the stages of clinical trials and on the science, combination immunotherapy, if we can get it to work even better in more people, better control, longer control, that I'd imagine, if I was going to predict the future, and I hate doing that in pure science because there are so many unknowns, I imagine that would reach the clinic first. I think there needs to be more coordination, globally. The IAS, the International AIDS Society, I'm currently president of the IAS, and I co-lead their Towards a Cure initiative with Tumbina Dungu from South Africa. And part of our mandate is to get that coordination across the world in several ways. First, on scientific strategy. So we publish a global scientific strategy pretty much every 4 to 5 years and try and get alignment of where people think the science is going. On capacity building and training, particularly for researchers in low and middle income countries. That's a big focus of ours, that more research needs to happen where the virus is, particularly sub-Saharan Africa. Fantastic, the research is happening in the US, Europe and Australia, but ultimately we want these interventions to work in low and middle income countries. And engagement in industry, because industry are a key partner here as well. They have the kind of scale and budgets to really, develop a product, and we have to be working with industry too. I think amfAR's investment in Treat Asia is a huge opportunity for cure research. At the moment, Treat Asia is largely an observation, very, very powerful, large observational study. Every country in Asia people that work in the field know Treat Asia, so it's a great brand that's been there for so long and done some great work. Treat Asia traditionally has not done basic science or clinical trials, but actually there's so much work needed in cure research beyond clinical trials and fundamental science. And part of that is just understanding what people living with HIV want in those countries, what they're understanding, their literacy around cure, clinicians' literacy around cure, observational studies in countries that have different strains of virus, or where many people living with HIV also have other infections like hepatitis B, TB, and others. All of that can impact the challenge of cure. So I think there's lots of really important groundwork amfAR could do through Treat Asia, which of course is the second largest group of people living with HIV, and a range of different capabilities. Some countries in the Treat Asia network have extraordinarily good capabilities in clinical trials, an example of that being Thailand. So I think it's a great opportunity. I've been having a few discussions with Annette Sohn and Rowena Johnson about what we could potentially do through Treat Asia. I'd love to see something happen there on the cure front. Lots of lessons that HIV can learn from COVID. Just give you 2 examples: the level of coordination and what was achievable when people worked together. Of course, that was supported by huge investments. I think it's unrealistic to think we'll ever get that level of investment in another disease, given the impact of COVID, but still the level of coordination and how people collaborated on multiple levels. And in the other areas, the science, I already gave you an example of mRNA and lipid particles. That field has exploded because of COVID, but there are other advances in COVID therapeutics that are highly relevant for HIV. One of those being antibodies, which are now licensed for COVID. We know how to make them better. We've got different ways to deliver them, and that's all through COVID. So I'm optimistic that the investment in science through COVID will have great impact on other major infectious diseases, including HIV.